All right. We all ready to go?
Yep.
All right, great. Well, thanks for the invitation to speak with you today. One of the few positives, I guess, to come out of this COVID pandemic is the increased availability of virtual forums for mentoring and conferences and talks like this one. And I'm particularly excited about chatting with you guys today because I've really spent my whole career partnering with clinicians and either of us can do individually. And that's a little bit about what I'm going to talk to today. I'm going to introduce a little bit of the work being done by engineering at Mayo Clinic, and maybe sometime I can come back and chat about what our normal work is. But over the last few months, we've really been doing most of our work has been focused around our response to the COVID pandemic, and that's what I'm going to focus on today. And most of you may know that the pandemic is gaining momentum again in the United States, and I think the United States may be a little bit challenged over the next few weeks, particularly with trying to send children back to school. We'll see where we end up. Where I live, it's the state of Minnesota. It's pretty well controlled and contained at this point, and hopefully it'll stay that way. So let me quick introduce myself. I'll just run through this quickly. My background is electrical engineering, biomedical engineering, and I've got an MBA as well. I started at Mayo right out of graduate school and spent about 16 years developing medical devices with a focus on sensors, electrodes, analog circuit design, microprocessor design, and of course, the programming that was associated with those embedded controllers. I've also done PCs and mini computers. So really, a lot of work, but all of it focused in the medical device area. But for the last 20 years, I've been in management. I've moved around to a lot of different management groups, managing electronics development, software development. I've even managed our mechanical unit for a while. And then most recently, about a year ago, I took over as chair of the Division of Engineering here at Mayo Clinic. So many of you may not be really familiar with the history of Mayo. We were founded in 1889 with a partnership between the Mayo brothers on the left, Will and Charlie Mayo, and the Sisters of St. Francis, which was a group of Catholic nuns in Rochester, Minnesota. So the Mayo brothers ran the clinical practice, and the nuns ran the hospital for almost 100 years before the two organizations merged in 1984. Mayo Clinic is the largest integrated not-for-profit medical group practice in the world. Really the first organization to organize itself around the concept of bringing the specialties to the patient instead of the patient going out to the different specialties. So, this was something that we put in place, early in the practice, and it is something that caught on all over the United States and eventually all over the world. So we consider this really to be the first invention of the Mayo brothers. So my group grew out of the rapid proliferation of surgical tools that occurred shortly after the development of anesthesia and aseptic surgery. So, anesthesia and aseptic surgery both showed up in the late 1800s, and that really changed the way we were able to perform surgery and really perform medicine. So initially, the Mayo brothers and their partners would create their own instruments, but eventually they realized that they needed a dedicated group of machinists to support the growing complexity of the surgical instruments. And so the instrument shop then was formed at Mayo in 1915, so we've been around 105 years. And here you see a photo from a 1920s article that described the manufacture of bone screws using beef bone and a lathe in our machine shop. So these bone screws were used in the repair of femur fractures, and similar devices were made for spine stabilization back in the early 1900s as well. In 1948, the shop was reformed as a division of engineering, so we brought engineering into the mix. And one of our first projects was work which on what would become the Mayo Gibbon heart-lung machine. So this was the first commercial heart-lung machine that was available in the world. And it's sometimes hard to believe that it wasn't until 1955 that we actually could perform bypass surgery. Other inventions at Mayo Clinic included the G-suit. This was developed in the 1940s by a team led by Dr. Earl Wood. It's one of our early aerospace engineers. Early patient monitoring was developed by our team prior to the establishment of commercial patient monitoring companies. So, I know you're all familiar with, the monitoring companies that you would see in intensive care units and surgical suites, but there was a period of time when there weren't companies making that stuff. And so, groups like our engineering group had to make our own. And so if you think back on kind of the development of electronics, it was the invention of transistors, which happened in the 1940s, the integrated circuit, which was invented in the 1950s, mini computers, so our ability to write software showed up in the 60s, and then the release of the very first commercial microprocessor occurred in the 1970s. So really only allowed us to develop new medical instrumentation. Also in the late 70s and early 80s, we assisted with the development and research on the first high-speed CT scanner. This was a dynamic spatial reconstructor, which was installed at Mayo in 1975. I've got to move the chat window over. So when we look at technology advances on a geological timescale, the era we live in today is really unprecedented. So if you look at the history of innovation, it took millions of years for us to go from the early tools to fire, and almost another million years to develop from hunter-gatherers to an agricultural-based society. And the changes just keep coming at us faster and faster. In the next decade, we're going to see technology that was only dreamed of. I still find it hard to believe that the iPhone is still only about 10 years old. And you think about the ways that that invention has changed. And it should be clear that, the next decade is really going to be interesting, I think. So my group, the Division of Engineering, our mission is to build unique medical devices to enhance patient care. We haven't been asked to save Mayo money by making things less expensively. And we haven't been asked to make things that someone else already makes. We've been asked to help create new devices in order to better serve our patients. Some of our work over the last 5 or 10 years includes early telemedicine systems, which is obviously very relevant nowadays. We've done extensive computational modeling of physiology. This was part of a project we worked on to help predict the risk of femur fracture in elderly adults. We've done some transformational surgical instruments like this device, which has since been commercialized. This allows the repair of the chordae and the mitral valve without having to resort to bypass surgery. In the early 2000s, this was a very simple 3D printed clip. It was actually our first 3D printed project, but it was to help repair it was for a commercial dialysis machine that was having issues with the tubing separating. We also have worked on a prototype bioartificial liver, which has sustained life now in animal trials for 72 hours. So our group focuses on medical device development. So we have an FDA compliant quality management system. In the US, that's based on FDA 21 CFR 820, but worldwide, what you're probably more familiar with is going to be ISO 13485. The US is going to be switching to that standard over the next year or two, so that we'll be a little bit more consistent with what the rest of the world is doing. Our group does do, full life cycle collaboration. So you come in with a concept and we prototype something quickly just to see how it works, all the way through to finished devices that you can take in and use on patients. So we provide prototyping support and then support for cadaver, animal, and clinical trials, limited manufacturing, and the creation of medical devices that we can use on patients. We have 67 staff, a dedicated group of management office almost 20 years ago. And then we have electrical, mechanical, software, biomedical, and chemical engineers on staff. I think we have one physicist. And then electronic technicians, mechanical designers, machinists, and we also have a scientific glassblower on staff as well. So I think, you know, all of us have been through kind of a tough, 4 or 5 months now. At Mayo Clinic, we started out on pace to lose about $3 billion after the onset of the pandemic. We had salary reductions, we had staff furloughs, and at the same time, we were really preparing for a dramatic decline in non-essential care and an increase in the COVID-19 cases. You know, we've since been able to come back from that. And part of what I'm going to talk about is how we were able to go back to doing elective surgeries and really being able to bring our patients back into our facilities safely, both for the patients and for our care providers. And, you know, we all have family and friends that were affected as well. You know, my sister is the nurse manager of ICUs at a hospital called UC Health in Colorado and has been dealing with this for the last 5 months. My niece is a new nurse, 1 year out of nursing school, working in South Dakota, where they had recently had a big outbreak at a meat packing plant. And they're currently doing a really interesting social experiment with a large group of about 250,000 people who are attending a motorcycle rally with no masks and very little social distancing. So we're going to see how that works out. And of course, this is a model of that little devil that's been causing us so much problems the last few months. So what I want to talk about is what engineering did. And so let me develop medical devices. So this is a long-term process. There's a, you know, it's a 2-3 year kind of development process. And we had to very quickly pivot because one of the things, I guess it's fortunate when you work at an organization like Mayo, everybody understands the value. Our values, they understand why we're here, and we are here to create a better experience for our patients. And so the minute the pandemic hit and we started hearing about supply chain shortages and, you know, ventilator shortages and things like this, we started looking at what we could do to help. And so there were a number of things that we did. One of them was around some of these supply chain issues. We did some reverse engineering of parts. We developed some alternate types of and sources for PPE. We developed a lot of aerosol and droplet mitigation type devices to reduce exposure to patients and staff. And we also did an interesting exercise to recertify our operating and procedure and clearance times so that as we went back to normal, we could cycle patients through a little bit quicker. And I'm going to talk about all of these things. So let's start with reverse engineering parts. So we're probably all aware of the stories that were coming out of Italy early in the pandemic. They were running out of disposable parts for ventilators and nebulizers, and there were these stories coming out about the creation of 3D replacement parts, and that really captured people's imagination. And of course, when the pandemic reached the U.S., you know, that was something that we had in the back of our mind because we had seen, you know, the challenges that the healthcare system in Italy had faced. So when the pandemic reached the U.S., our hospitals in the U.S. but the supply chain was bare. We really couldn't get a commitment to, you know, fittings and disposable parts for nebulizers and ventilators. And remembering the stories that were coming out of Italy, some of our staff came to us and asked us if we could 3D print these parts. And you can look at some of these parts and you can envision that, yeah, you could probably pretty easily 3D print them. So we went to talk to them. And 3D printing is really good for prototyping, but it's not so good for mass production. And when we found out what they needed was 5,000 parts a week, we really quickly moved to a different solution. So we ended up moving to an injection molding solution. It's one of the advantages that we have at Mayo relative to many other healthcare systems, that we have a full a full suite of engineering tools. So we aren't limited to a single tool like 3D printing. So we're able to use the right tool. So many of these parts we had print or molded in the thousands or even tens of thousands. So in the upper left here, you see a commercial package for a nebulizer, and this was something we simply couldn't order, and we needed tens of thousands of these. So on the left-hand side, you see a replacement fitting that we designed and manufactured ourselves. We also ran into supply issues for both PAPR hoods and happened to have about 9,000 obsolete PAPR hoods that wouldn't work with our new powered air purifiers. So what we did was we injection molded several thousand adapters so we could take these 9,000 hoods and actually make use of them. We were just fortunate that nobody had cleaned out the warehouse, and we still had all of these old air hoods sitting in boxes somewhere. We also developed a lot of new types of personal protective equipment for our frontline staff and our laboratory staff. So this was one of the early ideas that came out here, but I mean, there were people doing this all over. I saw pictures of this from Japan. I'm sure there were many places in Europe doing something like this. The idea was on the left-hand side is a simple intubation box. So this is a plexiglass box you can put your arms in so that you can do intubations, and the idea is to reduce exposure to droplets. And on the right-hand side is a shield that was developed for laboratory staff to use when they were working on infected samples. So we did a number of different barrier type devices. And here you can get a better look at that intubation box. And there were a number of different designs that were used to make sure that you weren't interfering with the procedure. And I'm going to come back to this one in a minute because there was a problem with this that was at the beginning of the pandemic, probably because we were rushing so quickly to try to find solutions to the problem. Another thing that we did was created over 20,000 face shields. So again, this was something that were in short supply in our supply chain. We couldn't get them. So we pulled in a group of staff, did a little bit of 3D printing, actually used transparency paper to make the shields, and made about 20,000 of these until our supply chain was able to get reestablished. Turned out the way Mayo reestablished our own supply chain for face shields was we worked with a local company to bring up an assembly line. So there was a manufacturing company nearby that was willing to convert their line to creating face shields, and they committed 15 or 20,000 a week to us. We also had a lot of requests and ideas that were coming in from staff, from external organization, even from friends and family. It seemed like everybody had an idea, and to be honest, almost all of them had something to do with 3D printing. And deciding what to do and what not to do turned out to be one of the really biggest challenges. And fortunately, Mayo has a really good incident command system that we engage in situations like that, and that was who we were able to turn to get our priorities and figure out what needed to be done first. Now, soon, I mentioned the PAPR hoods, the old obsolete ones that we were able to repurpose. We've also had a couple projects going on to develop our own PAPR hoods. So we're developing both PAPR hoods and the powered respirators that go with them. So as far as the hood goes, we worked with the sewing shop that we have within Mayo Clinic to develop prototypes for these hoods, which we are now creating our own supply of PAPR hoods. And then in addition, we've also been working to develop the powered respirators themselves. So it's a pretty simple design, and in fact, the powered respirators that are available on the market today are really an industrial design. They're not designed for a healthcare environment. So they're designed for a shop, for somewhere that's really dirty. The filters are designed to be replaced, and never get dirty enough. They need to be replace. So that allows a lot of efficiencies when you're designing specifically for that particular application. So that's what we're doing. We're developing these new powered air purifiers. We're working with a company out of Minneapolis, Minnesota, to create the filter material. So it's an N95 type filter material, and we will start providing these to our staff once we get them NIOSH certified. So NIOSH is the certification body in the United States that would certify air filtration devices. We've done other work in this space as well, looking at replacements for N95s. Early on, N95s were another item in really short supply. I remember early on in the pandemic, we had a confirmed order for 1 million N95s that was confiscated by the government, and it didn't show up. So we started looking for alternatives just to make sure that we were able to continue to meet our needs. So we've been working to supplement a couple different commercial half mask respirators with expiratory port filters. So these two filters, the one on the left is from Drager, the one in the middle is from 3M, and they're really good at protecting the wearer. But what they both do is they allow the expired air to come out from the respirator surgical field, for example. That's just not something that you want to do. So what we're doing is designing a filter, an expiratory port filter that will adapt to both of these types of respirators in order to filter the expired air. Here's one of our surgeons heading in for a 14-hour operation using the Drager mask combined with 2 other masks. It was a makeshift solution until we're able to provide her with an appropriate expiratory port filter. And she and her team are shortly releasing a paper on the tests they did to validate this use of the respirators, because they set up a series of tests to make sure that using them in this way was not going to put our patients at risk. Another thing that we've worked on are some kind of new and novel devices. So we're back to N95s once again. So that had got a lot of press early on in the pandemic was reprocessing or sterilizing N95s to make them reusable. So there was a paper that came out of the University of Nebraska in the United States that was using a low temperature system. So essentially using a low temperature oven for a period of time to reprocess or to sterilize the respirators. We also looked at using UV. This was proposed by both Stanford and Duke universities, and a number of healthcare facilities, at least across the US, set up systems like these to sterilize the N95 so that they could reuse them. We also looked at commercial systems, so something like a SteriAd system to use. And we happen to have about a dozen Zentex UVGI systems for sterilizing rooms. So these are essentially, well, they're not really robots, but you can wheel them into the room, you set them up and start a timer, leave the room, and it zaps the room with UV to kill all the bacteria and viruses on the surfaces. So a number of places have set up systems like these in a room to sterilize N95s. What we ended up doing was using a commercial BioQuil vaporized hydrogen peroxide system, because we could set this up in a shipping container off site. We could sterilize all the masks provided by our healthcare system. The hydrogen peroxide gas, will penetrate through materials. So it didn't have a lot of the challenges that some of the other methods of sterilization do, and we can do these in bulk. So since probably early May, we have been collecting and sterilizing all of the used N95s that are coming out of our facility. We haven't ever put any of them back into practice because we haven't had to, but we've got that in place as an emergency backup. We also started working with one of our cardiologists. So this cardiologist does a lot of research with electroporation. So electroporation is often used because it can facilitate the movement of compounds into cells. To move in and out quite easily. So it's a good way of getting medications into a cell. But if it's ramped up a bit, it can also totally break down the cell wall. So this team thought they could develop a device that could be used to sterilize individual N95 respirators, thus allowing a lab to reuse, or a person even, to reuse the same respirator over and over again. Because one of the challenges with mass sterilization is it's really hard to get the respirator back, the N95 back to the person that it came from. And this was just a little bit more appealing from the standpoint you could set it up in a lab. So to start with, we did some modeling of the electrical fields to determine optimum voltages and spacing for the device, and then we fabricated a prototype in our shop. And this work is ongoing. What the cardiologist envisions is kind of a vending machine like device where you drop your mask in, you put ready to go. So we'll see how that ends up. Another thing that we've worked on is a number of aerosol and droplet mitigation devices. And I had talked about the innovation box, the original innovation box at the beginning. That was kind of how this started. But we learned pretty quickly that those original innovation boxes had problems. And one of the you expose the room to a large bolus of aerosols all at once. So we quickly transitioned those to developing negative pressure boxes. And this is an example of one of those innovation boxes that has been turned into a negative pressure box, so that we're sucking the aerosols away rather than exposing people in the room. We also started modifying things like the standard face mask to create a barrier. So we added a negative pressure port to this to remove the aerosols before they could enter the room. We added ports to allow for endotrach tubes or suction devices to go in, put one-way valves on them, and a few things like that. It turns out that these devices are really, really hard to test because a normal person really doesn't give off much in the way of aerosols. And trying to measure the aerosols coming out of a person is really challenging. So it's hard to tell if a device like this is actually effective. So we ended up we had to get the number of particles in the air way down from baseline to even be able to hope to measure aerosols being released by people during different procedures, and then being able to tell if a device like this was effective at all. And it's fortunate we happen to have an aerosol science engineer on staff to help with that. We also did some simple disposable drapes like this. So it's a variation on that innovation port idea, but it's a disposable drape that you can unfold, throw over the patient, use, and then throw away. This one had a specially designed 3D printed access port on it. So very similar to the innovation boxes in function. We're also experimenting with larger boxes that will allow better access and more airflow to allow unimpeded access to the patient while still removing aerosols that the patient through Zoom. What I know is it's going to work on my side. I'm going to see a really nice video. I'll describe it. So the operator has got a little bulb that they're pumping, and what that is doing is releasing the side of the mannequin, and then you can see the airflow across the system. So the aerosols are quickly being pulled out before they have a chance to expose the provider or the rest of the room. Now the last part of the work that I want to talk about is probably the most clinically significant work we did, and this had to do with aerosol clearance and operating in procedure rooms. So I'm not familiar with what the standards in Europe might be, but in the United States, the CDC, the Center for Disease Control, has a set of standards that relate to the number of air changes per hour that need to occur in a room where an infection, an airborne or an aerosolized virus might be. And the standards are seven air changes before you can bring the next patient in. And so when we had to implement this protocol, because of course, the infectious nature and the aerosolized nature of the coronavirus, that really limited the number of patients we could get through a particular operating room or procedure room. And it was going to be really hard for us to get back safely to normal patient volumes. Now, in the US, these standards are about 20 years old. They were based to be generic, so they apply, basically all across the country to any procedure room or any operating room. And of course, what most hospitals use nowadays are very modern, highly efficient, highly effective laminar flow rooms. And these clear the aerosol out of the room almost as quickly as it's generated. So what we decided is that these generic guidelines needed to be challenged. And so that's what we did. And we knew when we went in and actually measured the performance of the rooms, there were three possible outcomes. One is no change, right? Nothing might change. The measured room closure time is very similar to the guidelines from the CDC. Or what we were hoping for and expecting is that we were going to be able to decrease the room closure times using the actual measured aerosol clearance time for a particular room. And of course, there's always the chance that the room closure time could increase. So that's always a risk you take. But for us, it was worth it because if the room needed a greater air clearance time, then that's what we wanted to do because we wanted to make sure that our staff and our patients were safe. So I'm going to talk to you a little bit through the process we went through. So we had over 600 operating rooms and procedure rooms that we needed to test, to characterize their aerosol clearance performance, so we knew exactly how they performed. So we had to develop a methodology. So we started by determining the room's baseline aerosol clearance capability. So we measured that for the room. Then we introduced a challenge aerosol and then monitored that aerosol concentration over time, and looked at how fast the room ventilation would clear the aerosol. And then we used that to calculate the new room aerosol clearance time. So how we basically did that, we started by doing something called a smoke test. So we would go into the room and we would release this puff of smoke. And then we would measure the background content. And the reason we were doing that is we wanted to move around the room. We wanted to find out if there were dead spots. The fear was, that this aerosol would collect in areas of the room and it wouldn't be effectively cleared by the room ventilation. So that was the first thing we looked at. And then we did the aerosol challenge. So we would put a large bolus of aerosol in the room, and then we needed to measure the clearance time. So the CDC standards in the United States are that the room air clearance time is the time it takes for 99.9% of the aerosols to leave the room. And it's an exponential curve, this air clearance time. So we had to characterize this curve by making measurements. So we released the aerosols into the room. We had a particle counter so we could measure the exponential decay. And that's what we use to determine the air clearance time in a particular room. So here's an example setup. And on the right-hand side, you can see an aerosol generator. That aerosol generator might look a lot like a humidifier because that's what it is. We had bought some commercial aerosol generators. They're readily available. The problem is our laminar flow rooms, the operating rooms, the laminar flow operating rooms were so efficient, we couldn't get a bolus of aerosol in the room. It was getting cleared off almost as quickly as it would show up in the room. And so we ended up going with a set of commercial humidifiers. We added a saline solution to it and used that to generate our aerosol, and that worked really, really well. On the left-hand side, you see a particle detector. This particular one is called a P-track. I'm sure there's other versions of that, but that's the version that we're familiar with. This is a handheld particle counter, so you can move it around the room, and you can check the particle concentrations at different spots in the room. Now here we have another video. This is showing it in action. Hopefully you're able to see the aerosols being released into the room. This is not an operating room. This is one of the procedure rooms. And here you can see the readings on the P-track from across the room. And now you can see the P-track readings stabilizing there. So we would take that data, we'd measure over time, we would generate a graph so that we were able to identify when 99.9% of the aerosols were removed. And the results we got looked something like this. So we started out, turn on the laser pointer here. We might have started out with a room clearance time, a posted room clearance time of 28 minutes, and our measurement in this particular case was 20 minutes. So we were able to recertify this room as a 20-minute room, not a 28-minute room. And as we went through, we had pretty good results all across the board. So you can see on the blue line was the standard clearance time based on CDC guidance, and the green is on average our measured air clearance time. And this is probably a little better chart for looking at it. This is a group of operating rooms, and you can see across this group there was on average a 40% reduction in the amount of time we had to leave a room empty. So the results, 80% of the rooms we were able to decrease the room closure time, 12% were no change. And this was kind of confirmation that what we were doing was working, because 8% of the time we actually found room closure times needed to increase. And it turned out that those increases were associated with a particular HVAC system configuration. And so that's something that our facilities engineers are now going back to look into, how to retrofit these rooms so that they're more efficient. It has to do with the way the cold air returns, the ventilation returns in the room were configured. One last thing that we're working on that I want to share, because I thought this was kind of an interesting study as well. We are working on a masking and physical distancing study. So what we're trying to find out is what is the effect of masking on the 2 meter or 6 foot recommendation for physical distancing. You know, we know that the droplets that are being expelled from a cough or a sneeze, they're impacted by gravity, so they're pulled down, and on average 6 foot seems to be, or 2 meters seems to be an appropriate distance, but we know under some situations they can travel further. So we're doing a study because we want to find out what happens when you mask. Masking will interfere with the movement of droplets. You know, the droplets are, you know, they're a heavier droplet. That's why gravity pulls them down. But they also have inertia. And when they hit the inside of the mask, they lose some of that inertia, that momentum. They still escape out the side, they escape out the top, so they're still showing up in the room. But the question we want to find out is, how far are they going under those kinds of circumstances? So we're setting up 2 mannequins. We're gonna try them unmasked. We're gonna try a variety of cotton and surgical masks. And I think we're also doing an N95 respirator. And what we're looking at is how far do these particles travel under those different conditions. One of the reasons we're doing this is because, you know, I think everybody is dealing with some of the same issues. How many people can you put in an elevator? How close can they be together? You know, when you've got staff that are working in areas side by side, how far apart do they really have to be when they're masked? This is really important information to have, because in all honesty, many of our facilities can't support, you know, 6 foot or more distance between people on a regular basis. And it's definitely our expectation that the use of masking would allow people to be a little bit closer together. But before we make any recommendations like that, we need to actually establish, test, and demonstrate that it's true. I'll show you one last slide. So this is what's behind the scene in the mannequin, is a relatively complex breathing simulator. So this device is going to simulate, you know, normal breathing, which is normal breathing is very low velocity, and it doesn't generate much of anything in terms of aerosols. And what we're concerned with these masks is not aerosols, but droplets. And there's very little in terms of droplets generated here. But then we're also going to want to simulate talking, which is a little bit more, and then coughing and sneezing, which are, of course, the activities that tend to generate the most droplets with the most energy, and thus, you know, traveling the furthest. So as I mentioned, when the pandemic started, we thought, yeah, it's going to be business for usual for us. We're going to go work remote, but we're going to keep doing the same kind of stuff. We never expected to be in the middle of addressing shortages of parts or PPE, and never expected to be working so hard to make sure that our staff and patients were safe during this. But the minute things started to go bad, we were looking for ways to contribute. And that's what happens when an organization has a compelling mission that everybody understands. Nobody told us. Nobody came to me and said, Mark, you guys got to go look for ways to help out. Nobody told us that. We just started doing it. And we're not going to be out of this for a long time, but at least at Mayo Clinic, our patients are back and our patients are safe. And we know a whole lot more about this than we knew, you know, 5 months ago. So I think even if we get more cases here, we now know how to manage them. All right, and that's what I've got at this point. I'm certainly happy to take any questions from you.
Thank you, Mark, for such a great insight. It was a great talk about the pandemic and how you got out of it, let's say, and how it is important to have engineering, science, and medicine together, working together for a better future. So we do have some questions for you. There is one from Banu. So you taking part in a very productive department. Were all creations initiated from clinical problems, or were they created after searching and wondering as an engineer spirit? That's the first question.
Yes, so they were coming from a lot of different places. So, for example, the ideas around intubation boxes and then the negative pressure devices that were used to remove aerosols in the rooms, those were coming from our clinicians, because they were the ones they were the ones at risk, right? They were the ones with patients at risk. They were the ones dealing with us, and they would come to us and say, I got a problem, can you help? Right? So it was our anesthesiologists and our GI folks and our pulmonary folks were probably the leaders there. But it turned out we happened to have an aerosol science engineer on staff by happenstance. Okay, so this was somebody who'd worked in the aerosol science industry for 15 years and then came to Mayo. And 15 years later, for the first time in his Mayo career, aerosol science was important. So we had an actual expert, because our physicians understood the problem and they were trying to do these makeshift solutions, but they didn't understand aerosols and how they move and how they spread and the difference between aerosols and droplets and how filtration really works. And so having that person on site was incredibly valuable. Now, it turned out that he also was planning on retiring on May 29th. And he had to delay his retirement twice while we worked through this issue. And as part of our plan to mitigate, you know, his eventual retirement, we contacted an aerosol science researcher at the University of Minnesota. So PhD mechanical engineer, one of the few aerosol science researchers in the world. Turns out it's a very small field. Who knew? So now we're partnering with him. So that was the stuff that was clinician driven. A lot of the supply chain stuff that I talked about was supply chain driven, right? So our command center, one of the first things they were doing was they were reaching out to respiratory therapy and some of the other clinical areas that use disposable parts that we knew were going to be in high demand if we had a lot of patients. So the folks with nebulizers and ventilators and things like that. And they started, kind of estimating, kind of worst case and medium case kind of risks and what kind of supplies they needed, and realized it wasn't there because they couldn't get it. So that's when they started asking us about how can we 3D print these? And they're like, can you 3D print this valve? And I'm like, sure, we can 3D print that. We can 3D print about 12 of those a day. How many do you need? And that's not going to meet their 1,000, 2,000 a day that they need. So that was another source of projects, and was a supply chain. And then that's how we got tied in with the PPE as well, because it was there with most of the PPE. So the PAPRs, the PAPR blowers, the respirators, surgical masks. We're working on developing surgical masks for a while. These things were all things that were normally you would just order them. And so supply chain could tell when they weren't going to be able to get a hold of them. Face shields, that was another big one. Just couldn't order them. Nobody would guarantee a delivery. So that was the other group that we worked with a lot.
Okay, thank you. Another question, 3D printers are not exactly a standalone solution. Thanks for mentioning this. You need to get the right combination in order to go beyond the prototype. What other methods are there to produce the digitally obtained other than 3D printing, injection molding?
Yeah, well, I mean, there's a lot of standard manufacturing techniques, right? So 3D printing and injection molding are two, but we have a full machine shop, so we can machine pretty much anything out of metal. So there's both additive, we call it additive and subtractive manufacturing. So additive manufacturing is all the 3D printing kinds of things. Our group has several different polymer printers that can print in a number of different types of polymers. We also have a metal printer so that we can print metal parts as well. Maybe I'll come back sometime and talk to you about our quest to start metal printing patient-specific implants. That's a kind of an interesting effort we have going on. So the other thing that we have is many of our engineers have extensive manufacturing background, having worked at other companies, and so other than the manufacturing capabilities we have in-house, they're very familiar with kind of some of the commercial manufacturing options that are out there. And
Sorry. Another question that we have is, do you think that situations such as pandemic that put people in a difficult situation and suddenly develop are the turning point and encourage researchers to produce more creative and effective work?
Well, that's a good question. One of the things it's done for our group is it's really focused us on how to deliver something safely fast. Okay, and honestly, when you're developing medical devices, it's easy to spend too much time working on the process and do some things that aren't necessarily value add. And this forced us really to take a close look at those things, because it's a challenging discussion. When you have something, you need to make this really fast because people's lives are at stake, but the device itself has risk, right? I mean, that's a really tough challenge to balance. So we actually got a small group of us and went off site for a few hours and actually went through our process and said, what do we need and what can we jettison and how do we do these things in parallel, and how do we still done the way they need to be done, but they don't take so much time. And that's something that at least our group is going to carry with us when we go forward. We're going to leverage what we've learned to really become more nimble and more agile. And I got to tell you, this is really tough for our safety folks. So if we have some dedicated safety engineers, and to be a safety engineer, you really have to care about the details, and you agonize over you know, very small things that can make a big difference. And so there's a balance, right? Is the risk of using the device, right? Is that risk greater than the risk of not using the device? So the device could cause harm, but not using it, it can also cause harm. And so where is that balance?
Thank you. We have a question from our participant, Amar. Where can we learn or read more about the specific solutions you talked about? For example, the mask sterilization devices and distancing studies. Are there any published data?
There is, and I don't have reference to them offhand, but there were a number of universities that were working on this stuff. So, for example, there's papers from the University of Nebraska talking about heat sterilization, low temperature heat, which was about 160 degrees Fahrenheit. I don't know what that would be in Celsius, but something under 100, so not terribly hot. Something you could do on a small oven. Was Duke and Stanford that had UV sterilization systems. UV sterilization works great for the mask. You can reuse them maybe about 5 times, and this was published in the study. They looked at reuse as well, because UV will deteriorate the material. The challenge with UV, of course, is that it only disinfects line of sight. So if you have something that's covering something else, it won't get behind. It's called a UV shadow. It has no penetration depth. So, you know, that was one of the reasons we didn't use that. There's even articles out there about people suggesting you take 5 paper bags and you mark them Monday, Tuesday, Wednesday, Thursday, Friday, and you just put your mask in it, because, you know, what's the shelf life of the coronavirus was maybe 5 or 6 days. You know, that was another option. The system we ended up going with, hydrogen peroxide, there's a lot of data out there on that. There was a company, Draper Labs, actually in the United States, has a commercial system based on this that they'll bring on site. A couple of articles about it, and then probably the best reference is so 3M makes most of the respirator, the N95s worldwide. They had an article out on various means of reprocessing them. They said we can't recommend them, but they were nice enough to provide the different ways to go about it and the different challenges you might have to overcome. And if somebody's really looking and can't find that stuff, they can certainly send me a note. I'm sure I've got copies of it stored and whatnot. So.
Thank you. Well, a following question just from me as a woman, but also as a scientist. We have this small UV lights for nails, you know, and also in the labs we have the hoods that have UV. Can we use them for sterilization of our masks? Do you think so?
Maybe. I mean, so that would depend on the strength of the UV. So you've got a UV source, right? And there's a certain exposure limit. It's that you have to obtain. It's given basically in lumens. It's intensity of the light for a period of time. So the closer it is, you know, the less time it has to be there. But you have to take into account the intensity and the time, right? So can you just use any old UV system? Well, maybe if you do the calculations. However, there is probably a lower limit. So if the UV is below, you know, some limit, it probably doesn't matter how long it's there. So I'm not going to say go do that. I would be a little bit nervous myself. I think you'd be better off taking a look at the Nebraska paper and sticking it in your oven for half an hour at 160 degrees myself. That seemed pretty promising.
Okay, thank you very much.
Mm-hmm.
So how do you see some of these solutions to the specific problem of the pandemic progressing to address other problems? For example, mass sterilization addressing a problem in medical waste and sustainability?
Yeah, that's a good question. I think what's going to happen is people are going to stockpile more stuff and it's going to lead to more waste as opposed to going the other way around. However, um, we did get very interested in those reusable half mask respirators. And as I mentioned, the two that we've looked at, one is from Draeger and the other is from 3M. The Draeger one is kind of interesting. It's an N100 mask, so it's even better than the N95. And you never have to replace the filter material. So it's basically reusable forever. It's a little bit bigger, right? It's heavier, it's a little more expensive. It's for one person. You have to go through and you have to clean them, disinfect them between uses. So, it's not quite as efficient as just taking your N95 off at the end of the shift and getting a new one the next day. So there's some trade-offs, but I know that our staff who are using them love them. And the woman I showed, that's Dr. Brittany Howard, she does these long procedures. And she's talking about, how wearing this N95 for 14 hours, how hard that is. And, and so these respirators apparently for her at least are a lot more comfortable. They get a better fit. I don't think they have to be on as tight. So it was interesting. We were talking to her and asking about, hydration. Would it be useful to have a hydration port added? She's like, no, I go into my 14-hour day already dehydrated, because she says, what happens if I have to take a break? She's like, I got to take all the PPE off and I got to put it all back on. She's like, no hydration.
Okay, thank you very much, Mark, for being with us today and for giving such an amazing talk. And of course, I would like to thank Clinician Engineer Hub and EIT Health alumni that co-organized this event, and everyone who has been in with us today. And we welcome you to join us in other talks that we are organizing all year. Thank you very much again for everyone.
And thank you again for inviting me. I certainly enjoyed it. I mean, I love the partnership between clinicians and engineers. So, yeah, a lot of great questions.
Thank you. Have a great day.
Bye-bye, all.
Bye-bye.